Right, so tonight, our title, as you all know at this point, is Exploring the Belowground Forest Ecosystem. We're talking about connecting symbiotic fungal networks with forest resilience through close to nature management. And we have been supported in this by the Department of Agriculture, Food and the Marine. So you're going to hear tonight about 30 years of research into the underground networks that support trees. You'll learn how CCF supports and maintains those networks. So with no further ado, I will move on. So the agenda for the night will be a quick introduction, which I'll be making. Then we'll have Liam from ProSilva telling us about ProSilva. We'll then hand over to Jonathan for the main meat of the night, and then we'll have some questions, general discussion, and a wrap up. So if you put your questions, you've all got the hang of the chat now, which is great. So your questions and comments into the chat. I'll be moderating that as we go along. I won't be interrupting Jonathan until the end, but I will be compiling questions for him to answer. And I suspect we have some other experts on tonight, so maybe they'll be able to help us with some of the questions. Okay, so who do you have here? So my name is Anna Brown. I'm a forest owner. I planted a forest in Kilteal, County Kildare in 1995. So at this point, it's doing very well. It's in its second thinning. I'm also, I have a master's in e-learning, so that's why I'm driving the webinar here for, I suppose, for both reasons. But I'm very interested to hear what Jonathan has to say. We also have Liam Byrne, who's the chairperson of ProSilva. He's a woodland contractor and sawmiller, and he's the one who actually does the thinning in my forest. So he's very good at what he does and has a deep knowledge of forest ecosystems. And finally, with Jonathan Spazzi, which we're delighted to have, talking about the mycology. He's a forestry development officer with Teagasc. He's very passionate about mycology. Studies and practice have focused on close to nature management and forest mycology in Ireland and in Europe. It was worth mentioning, because ProSilva is part of a European organization, we do a lot of work with Europe. So that's sort of, I'm sure Liam will touch on that as well. But John's got a lot of information he'll be sharing with you tonight, and we're very lucky to have him as part of the ProSilva community. So I'm going to hand over to Liam at this point.
Thank you, Anna, and you yourself are a much valued member of the ProSilva community, and thank you for your technical support over the last 2 years and bringing consistency to innovation and consistency to our online events. On behalf of ProSilva Ireland, I would like to welcome you all to our webinar. Firstly, as Anna said, I would like to acknowledge and convey thanks to the Department of Agriculture, Forestry and the Marine for supporting this event through the Forestry Support Fund. I would particularly like to welcome our valued members. To those of you who are new to ProSilva Ireland, I also extend a warm welcome. ProSilva Ireland is an all-Ireland organisation that promotes continuous cover forestry, also referred to as CCF, and the adoption of lower impact silvicultural systems. CCF aims to integrate multiple forest functions while maintaining a permanent forest structure. These functions include protection of soils and water, enhancement of natural habitats and ecosystems, promotion of diversity and resilience, enables carbon sequestration, and all this while providing the opportunity for the production of quality timber. In summary, the ProSilva principles supports forests that are both ecologically and economically sustainable. ProSilva is a not-for-profit voluntary organization founded in 2000 and is part of a wider international network of ProSilva organizations across 27 member countries. We enjoy diverse and dynamic membership of foresters, forest owners, contractors, ecologists, wood engineers, scientists and artists and many others. And I think we see that representation here this evening in the attendance. General activities include field days, international study tours, training, publications, and engagement with forestry and other interested stakeholders. There are many organizations supporting the development of forestry and woodland management, but I think what makes ProSilva unique is the international dimension, and being able to share vast experiences through international collaborations, study tours, visiting guests, sharing of publications and training tools. So to this evening, I'm delighted to have fellow committee member Jonathan presenting. Jonathan hardly needs little introduction to many of you, but to those of you joining from Ireland and beyond for the first time, Jonathan is an innovative forester with international experience fueled by both great curiosity and enthusiasm. Jonathan has worked in many aspects of forestry as a private contractor and now works as a forestry development officer in Teagasc. In particular, he is well known for his work in and great love of broadleaf silviculture. What has always, I think, distinguished Jonathan is that along with his silvicultural knowledge, he has always maintained a great understanding of the whole forest environment, including mycology. Tonight, Jonathan is going to share his knowledge of the below-ground ecosystem and its relationship with silviculture. So thank you, Jonathan, for giving your time to prepare for this evening and present.
Thank you, Liam, for the humbling and nice introduction. I'll try to share my screen, and welcome to all our viewers at home as well. Well, again, thank you, Anna and Liam, for the lovely introduction, and welcome to all our viewers. I'm really delighted to have this chance to talk about this subject, the below forest ecosystem, or the forest underneath our feet. I think this is a very important aspect of forestry, and one that perhaps has been neglected in forestry to date. So this is a very vast area, as many of you might be aware, so it's quite overwhelming to tackle. So we clearly won't be able to cover everything, but I hope that this presentation could act as a conversation starter for future, in the near future. So the relationship between fungi, forests, and trees, I might add, trees and people, has been a very important part of my professional and personal development throughout my life. My roots today are in the hills, lakes, and forests of East Clare, but my origins are in the Italian alpine forest and its forest culture, and in particular, its strong fungi foraging culture. In the picture here, you see my ancestral family village, and some of my early childhood memories would have been involving foraging fungi with family and friends. And this is a culture that I've tried to share with my family, with my children and friends. And this culture, to some extent, has been really influential in me pursuing a forestry career, forestry studies in Italy, North England, and Wales. And during my study, my formation, and my early practice as a forester, I would have experienced what I have considered important research discoveries in the last 30 years in this area. And I'll try to share those with you. For me, it certainly has been a very rich journey of discovery, and one that it continues to be. And in many ways, it's a journey that for all of us has just begun. So I think it's fair to say that today forest fungi has gone viral. Many of you would be aware of this. We have a rich forest fungi are now part of a rich popular culture, and we've seen in recent years that many successful books published on the subject. I have a couple here, just the Entangled Life, Finding the Mother Tree, but also many videos, many articles, and we even have a Netflix documentary on this subject released on Netflix recently. So while on one side we have seen in recent years some skeptics and even deniers to new theories, on the other side we've seen a more fantastical representation of new research. So what I'll try to do here tonight is trying to highlight a few concepts, present some recent studies, and really trying to reflect on how the findings, this research finding, could be incorporated specifically into what Liam referred at the beginning, a close to nature management approach. So we'd frame it within that. So the general structure of the talk, around 40 minutes, 30 minutes, 40 minutes, and I think I'll have it within that. Let me know if how we do time-wise as I progress. So just a summary of the general knowledge. I'll present some key research that I found significant over the years. We look at close to nature management in the context of the fungal network and reflection, and then we'll lead to question and discussion at the end. So I think to better understand forest fungi today and forest ecosystem for that matter, I think it's useful to take an historical and evolutionary perspective. It might surprise a few, but sort of taking a thousand steps back to take 1,001 step forward. So if we could go back 450 million years ago, we would see, or at least this is our understanding, that all lands were soilless and lifeless. Lands were barren wasteland of rock and gravel back then, and life thrived only in the oceans. The first life forms that emerged from the ocean to colonize these lands were in fact very similar to what we have today as lichens. So lichens are very interesting organisms, and they're made by a partnership of algae, carry out photosynthesis, capturing the energy from the sun, and provide anchorage of fungus that extract minerals from the rock and rain, and bacteria would work in partnership with the fungus. If we fast forward 450 million years to today, what we'd find is that this partnership is still really the foundation of all land ecosystem, and especially forests. The net results are rich forest soils, where trees, fungi, and bacteria relationship function as the engine of the ecosystem, also supporting a vast soil biota, from microscopic organisms to larger ones such as earthworms, etc. Which if we look at it in this slide, we'll realize how a forest soil in itself is a living ecosystem. And importantly, it's important to note that there's a lot of talk about forest carbon, and the soil is being mentioned as one of the important store of forest carbon. And it's important to highlight that a significant portion of soil carbon is actually in the form of living carbon, in other terms, the soil biota itself. So for some time, fungi were considered to be plants, primarily because they were understudied, and they have some similarity with plants. They grow from seed-like structures called spores. They don't move, but unlike plants, they cannot generate their own food, so they don't carry out photosynthesis. In fact, they source their nutrients like animals. They carry out digestion. They digest their food externally in the environment through acid and enzymes. The reality is that fungi are neither plants nor animals. They have their own independent kingdom, the kingdom of fungi. They're unique organisms with their own kingdom. Here you can see an Amanita muscaria fly agaric. So fungi come in different shapes and forms, and also come in different modes. They're not all made equals. They can be parasites in the sense they can actually kill trees. They can function as saprophytes, meaning they're able to decompose dead organic matter, being dead wood or leaf litter or twigs, or they can be symbiotic organisms. And this is the mode that we'll be focusing on more in this talk, the symbiotic fungi. If we take them all together, and they do in fact collaborate, all the three types, but if we take them together, their ecological function can be described as an interface between life and death by recycling all nutrients. In fact, fungi are able to break down all organic substances through very powerful enzymes and acids, and partnering in this with bacteria in order to recycle nutrients that otherwise will be locked away from plants, and with specific attitude for phosphorus, which is a key nutrient and hard to get in most of these land-based ecosystems. They also carry out important pathogen control service, water regulation, and bind all soil particles. In the pictures here, we can see some of my local species. This is a Pleurotus austriatus oyster mushroom in my local Rostora woodland in East Clare. Here to the right, a Colibia species in Littering Forest, which was actually the location of the first woodland festival in Ireland. Both saprophytic species, decomposers. So is the Lepista nuda bluet, very good edibles. In the symbiotic, just a few examples: Cantarellus shibarius, Chantarella, and Hydnum repandum, the hedgehog mushroom, all excellent edibles as well. And then we have Suillus granulatus from a forest nearby me in Clare. And finally, Boletus edulis, the world famous porcini, probably one of the best gourmet varieties, again back in my local forest here in Rostora, East Galway. So with all these flamboyant color, shapes and flavors, we might be forgiven for thinking that what we're looking here in this picture, for example, is the real organism. This is a Cortinaria subtortus in the forest just next door to me. In fact, what we see in this picture is the reproductive organ of a very clever and sophisticated organism that we call fungus, or the mycelium of the fungus. This organism is very secretive, often hidden from us, and this is probably one of the reasons why we are late to study them, but it permeates and facilitates all life processes. It takes the form of a network of living fiber, as I mentioned, called the mycelium, which is right under our feet throughout the soil. Is often invisible to our naked eye, and is estimated that 1 cubic inch could contain up to 3 miles of mycelium thread. So it's definitely there in a mature forest soil, in a healthy forest soil. Now, the mycelium of symbiotic fungi, it is directly connected with the roots of trees, as you can see in the drawing. In fact, each tree can be connected to several mycelium mats, and each in turn connected to several trees. This leads to generates a very interactive and complex network, and we are trying to unravel here tonight. So the tree fungus relationship is called mycorrhiza. And mycorrhiza is a relationship between a symbiotic fungi and trees' roots. It's a mutual exchange. So carbohydrates are shared by the tree in return for nutrients from the fungus, specifically phosphorus, but other nutrients as well. Which obviously boosts significantly the ability of a tree to feed. And ultimately, it's really a requirement for most trees and plants in order to thrive and survive. It's sort of an evolutionary strategy, you could describe it. Fungi also provide disease control through the secretion of antibiotics and antibacterial bacteria in the soil. If you could think of penicillin being extracted from molds, which is a type of fungus, and also provide water regulation through their mass. And in this sense, they can be considered the foundation of forest resilience. In the picture, you can see a seedling of pine, and in dark orange, its actual root system. The white network is the mycelium of a Suillus species. And you can see the obvious benefit to the seedlings in its ability to reach a far greater area of the soil. But furthermore, is the capillarity that the mycelium offer in terms of accessing soil particles. And if you factor in the ability of the mycelium to break down organic matter through acid and enzyme, you really get the full picture of the importance to the tree growth of the mycorrhizal relationship. This is a research from 2008, really showing how far fungi can go in their search for nutrients for trees and plants. And it would appear that are actually able to mine through rock using powerful acid to extract valuable nutrients. And researchers in this case believe that the black channels you see in the top left pictures are tunneled by fungal mycelium into rock particles. The bottom left picture is a magnified picture of a mycelium hyphae entering a rock particle. So to make things more complex, if they weren't complex enough, we have different types of mycorrhizal networks in the forest. The dominant network is called the ectomycorrhizal network, and is shared by the majority of forest trees and shrubs. And you can see a few listed here in the EM trees, ectomycorrhizal trees, trees like oak, beech, sweet chestnut, birch, pine, spruce, larch, Douglas fir, alder would all share on the common network. Other species like willow and hazel would also be, as a longer list, is a short list. However, a small number of trees share on a different network, which is in common with herbaceous plant called the arbuscular mycorrhizal network, AM. And trees like cherry, sycamore, ash, elm, holly, and yew would share on this network. One of my observation here is that those trees tend to occur more as isolated trees in a natural forest composition. I'm thinking here mostly cherry, the sycamore, and yew in particular. Tonight, we will focus on the ectomycorrhizal network primarily. Also, again, more complexity in there, while many fungi, symbiotic fungi species, ectomycorrhizal species are generalists, so they will be able to associate with most of the ectomycorrhizal trees. Some are specialists, like for example, the Suillus luteus, the slippery jack, which is only found in association with pine. So clearly, tree species composition in a forest is very important to determine the type of ectomycorrhizal network. So with all this said, we look next at a few studies over the period 1986-2019, the last 30 years, these are studies that I have found significant in helping us and me to better understand the EM fungal network. Each study set out to answer a different question. So starting in the mid-80s, we knew at that point that trees exchange nutrients with fungi, but there were key questions still to be answered, and specifically, can trees exchange nutrients across the ectomycorrhizal network? And if so, can transfer through the EM network can it be multidirectional? If that's all true, how does this work in an old growth forest? In other words, a direct study in an old growth forest. What is the fungal diversity of current Irish forests? And finally, closer to ourselves to today, what controls the flow of nutrients within the complex fungal network? I'll just breeze through all this different research. I won't get too in depth, but just to highlight the progression of the level of knowledge and understanding that has happened in the last 30 years, which I think has been very significant. So starting in 1986 and late mid-1980s, we knew then that trees exchange nutrients with fungi. By Finlay and Reed set out first to see if trees could exchange nutrients through the actual network. So they used seedlings of lodgepole pine and Scots pine in pots, inoculated with Suillus bovinus and Suillus granulatus, which we'll see in a minute. This is the seedling of pine infected with Suillus bovinus 2 weeks in. This is the same, another seedlings 5 weeks in. So what they did, they exposed one of the seedlings to carbon-14, and what they found that the label carbon-14 found its way in other seedlings, which was the first time there was showing a transfer of nutrients across the ectomycorrhizal network between trees. They also interestingly noted that if individual seedlings were shaded, the transfer of carbon was greater. Here, just a note of interest, this is the main actor, is the Suillus bovinus, which I encounter in one of my forest travel, which in this case is providing an important power nap habitat for this salamander that didn't look too amused with me at the time. So taking from Finlay and Reed finding, a young British Columbia researcher named Suzanne Simard set out to further study with colleagues the carbon transfer, this time between seedlings of birch, Douglas fir, and western red cedar. Instead of pots, they moved into a forest site with planted seedling, and they used isotope carbon-13 and 14 to label the transfer. And they discovered a multidirectional transfer of carbon, this time between the ectomycorrhizal birch and the ectomycorrhizal Douglas fir. The western red cedar, who's not an ectomycorrhizal species, but is an arbuscular mycorrhizal species, did in fact not share on the label carbon, which further confirmed the theory that the EM network was responsible for the transfer. So as in the previous research, the stronger the shading, the greater the transfer to the shaded seedling, which confirmed this idea of a source-sinks relationship between the parties. So this seems to be a transfer from high concentration to low concentration. This was for the first time suggested something quite different. So suggested that perhaps mutualism, not just competition, is a co-driver to ecosystem functioning. This study was published in the Nature magazine in 1997, where the editor decided to include it in the cover with the title "The Wood Wide Web." The Wood Wide Web, which this was the first time that this term, which is now commonly used to describe the EM network, was used. This article brought EM fungi to the global stage and generated quite a strong reaction, both in favor and against, with many skeptics and even deniers that this could be actually happening. And this debate still continues today. I experienced a little bit of this myself a few years back as part of publishing my one and only peer review paper on inventory protocols on close to nature management. And as I referenced Simard work in my paper, I remember one of the anonymous expert reviewers, they were reviewing the publication. Their comment, his comment was, sure, this could not be true. So it's true to say that much of past ecosystem theories have been based on the premise that competition for resources is the only driver. So it's quite a departure. It's quite a shift to consider mutualism as a possible co-driver. So following from Seymour's work, a team of researchers in British Columbia University, they set out to further explore the mycorrhizal network, but this time they moved into the forest, into a multi-age old-growth Douglas fir forest, and they uncovered an extensive network where they found that a large number of young trees were connected to the larger, older veteran trees. So they're confirming again the idea of a sourcing pathway, so nutrients would flow from abundance to scarcity in the forest ecosystem, mediated through the EM network. So the old vigorous trees seems to feed the younger, weaker trees through the EM network. So this is the origin of the mother tree concept that led to the title of the very successful book by Susan Simard, Finding the Mother Tree, that some of you might be familiar with. Now here's the final consideration from this research. To ensure that old-growth Douglas fir forests remain resilient and self-regenerative following disturbance, our findings support a management approach that conserves large trees or groups of trees and their mycorrhizal fungal associates. So we move on in our understanding. Coming closer to home, in the late 2000s, Coford in Ireland funded a 3-year national survey of forest fungi diversity, top left there. I was part of it as a field mycologist, and during it I surveyed permanent plots in a range of forest types in the Midwest. And it was a unique experience for me to be able to monitor closely the behavior of fungal species fruiting in the same location over a 3-year period, and really gave me a great insight into forest ecosystem functioning, and is an experience I'm very grateful for. So some of my own personal considerations on that experience is that I found, I sort of already knew it, but I confirmed that Irish forests are very fungi rich, perhaps due to the ideal climate we have. The fungi diversity and abundance appeared linked to tree species and tree vigor. And the mixtures of EM tree species with vigorous trees seems to do better. And also this might come as a surprise to some, as the sitka spruce actually behaves as a very strong ectomycorrhizal species. This particular observation was also the finding of a parallel study, which you see here on the bottom left, which was published by Richard O'Hanlon and Tom Harrington in 2011. And I read here, the result of this study have shown the Sitka spruce forest in Ireland can support a high fungal species richness. Finally, closer to today, a team led by Toby Kears in the University of Amsterdam, using new advanced techniques such as quantum dot technique and high resolution video have set out to observe the nutrient flow within fungal networks. And their conclusion are that fungi gain benefit from host plants by moving resources from rich to poor patches. Again, this idea of an equalization or a transfer from abundance to scarcity. And fungi in this sense act as nutrients traders within the ecosystem and control the nutrients economy. I think is an interesting concept, the nutrients economy in the soil. This is a very interesting video showing actually live the multi-directional flow of nutrients. You can see nutrients flowing in both directions, apparently very chaotic, revealing the complex and constantly changing dynamics at play, of which we have very little understanding still. Enter close to nature management. So Liam touched on this already. It is an integrated management approach. The aims to integrate social, economic, and environmental values at the forest level. This management approach has been supported really by our better understanding of forest ecosystem functioning and is increasingly supported by European and national sustainable forest management policies. So there's a lot of policy driving close to nature management. It's based perhaps on 4 key points. It is modeled on forest natural evolution phase, succession stages. It's focused on promoting individual quality trees through selective management. Harvesting patterns mimic natural disturbance. And ultimately, it aims for multi-species irregular permanent structure as a key really to provide resilience, biodiversity, strong ecosystem services, including, of course, sustainable timber yield through a production conveyor belt, so continuous supply. And next, I will look at a few typical features of close to nature management, in particular mixtures, tree mixtures, the presence of large, vigorous, mature trees, natural regeneration, and deadwood retention, and how those might interact with the ectomycorrhizal fungal network. So mixed species is a typical characteristic of close to nature management. In some cases, you might start with uniform plantation, but through the transformation process, we see a rapid increase of species diversification. From what we discussed earlier, it's easy to see how mixtures are likely, tree species mixtures are likely to strengthen the EM network. A higher number of tree species support a greater number of EM fungi, including specialists and generalists, so leading obviously to increased productivity and resilience. However, we have to be mindful that all tree species support EM fungi. We have arbuscular mycorrhizal trees as well as we mentioned. So also, as in the case for trees, fungi species follow a succession roadmap. So some ectomycorrhizal species are early starters, others only come in later on in the forest development. So early starter fungi are particularly important in order to kickstart the whole network. And I think there is here an overlap and a synergy between pioneer EM fungi and, species and pioneer tree species. And I mentioned one for all birch, being a very good species to transition what is a EM network in grassland, perhaps to an ectomycorrhizal network. So the inclusion of such pioneer tree species, especially at planting stage, is very important and is likely to boost the fungal network, leading again to greater productivity and resilience. Vigorous mature trees, a big feature of close to nature management over a transformation period, and studies, as we mentioned earlier, have shown that dominant large trees really disproportionately contribute to the development and maintenance of the EM network. So clearly favoring from early stages healthy individual, like the tree in the picture, for example, to develop into hyper vigorous trees as part of our irregular structure is likely to strengthen the fungal network, being for timber production or equally for cultural and social values. In any case, large trees are provide a great strengthening service to the EM network, again leading to increased productivity and resilience. In the picture here, you can see my daughter on her birthday on our local celebrity tree, the Brianborough tree. This is an ancient sessile oak tree who gets plenty of regular visits, and is supposed to have been planted by Brianborough himself, the last king of Ireland, 1,000 years ago, all of which is of course true. So, as mentioned, close to nature management is modeled on forest natural succession phases. So forest, as you see in the picture here, evolved from establishment stage to a young pole stage. As trees mature, start producing seeds, and from here, seedlings, at which point disturbance, for example, windthrow or harvesting perhaps, would facilitate renewal, diversity, and productivity within the forest. So given an irregular structure, the pattern in the forest that emerges from the regeneration stage is of one of scattered patches of regeneration, sometimes referred to as the waiting room that you can see there in the red circle in the picture. The seedlings here remain stored for a number of years, depending on species. Some species could be there virtually forever. Douglas fir and silver fir, for example. Others, not so long, a number of years. Oak would not as long as Douglas firs, for sure, but is still able to respond to light increase following onset of disturbance, again, being natural or harvesting. And you can see in the blue arrow here in this drawing, the removal of the harvesting tree is actually creating a gap that is facilitates then the release of a vigorous young pole tree, sometimes called a sprinter. Now, too big of a gap seems to give competitive advantage to vegetation requiring expensive management. If the gap is a measure size, we see successful release of sprinters. So the question, this is what we see actually above ground. This is the experience of close to nature management systems. So the question here is, what is the ectomycorrhizal fungi role in this process? So to answer this question, I might go back to the original paper from Finlay and Reed all the way back in 1986, about 38 years ago. And I read, in the forest, the majority of seeds of a given tree fall within close proximity of the parent, and those germinate in an environment already containing mycorrhizal roots supporting compatible fungi. Naturally, regenerating seedlings can spend a number of years in a suppressed condition below an adult population before opening of the canopy permits sustained positive net assimilation rates. Survival during this period, when the plant is living below its compensation point, might well be dependent upon sustained or seasonal supply of carbon from the illuminated overstory. In this way, seedlings are potentially able to receive mineral nutrients absorbed from soil at a considerable distance from its own root system. This was in 1986. Other researchers, as we have discussed, seem to have come to similar conclusion, and if we consider this hypothesis to be true, what we experience in natural forest dynamics and in close to nature management alike perfectly match this description. For example, one early close to nature practice was the cutting of holes in forest ahead of regeneration, hoping that the regeneration will come. This in general turned out not to be a good idea, resulting in loads of vegetation in the hole, in the gap, and not a lot of regeneration and loads of expensive management. One explanation for this would be that by cutting a hole, ahead of the regeneration, the connectivity between the ectomycorrhizal network and the gap itself is broken, and the gap in the gap, so giving way to arbuscular mycorrhizal networks, leading to an explosion or herbaceous growth. Instead, the opening of small diameter or relatively small diameter gaps within existing advanced regeneration have been shown to give best results, especially when coupled with diffuse light from an open structure forest. And this is a key point in close to nature management, the control of the structure and creating an irregular open structure. One reason for this is probably self-evident, is that it allows for the EM network connectivity between seedlings in the gap and larger ectomycorrhizal trees in the surrounding forest. Larger gaps are possible, and in that case, retention of ectomycorrhizal trees and shrub is recommended and would be beneficial. Studies have shown that tree fungi roots are likely to extend from the surrounding forest, not a lot, 8 to 17 meters into a harvesting gap, which seems to match, at least in my reckoning, in my experience, best practice gap regeneration in under close to nature management. In conclusion, I think a high edge to area ratio seems likely to maintain a high ectomycorrhizal fungal connectivity in this case, allowing for vigorous growth through a gap regeneration system. And so giving the saplings in the waiting room, as we described, the best chance to transition into few vigorous sprinters, as we see in the picture there. Deadwood is a big feature of conservation management and equally or close to nature management. Deadwood retention in all its form is often associated with insects and invertebrates and their diversity, but equally it's very, very important for fungi species, for fungal diversity, especially for saprophyte species. I would also equate this to our own nutritional requirement. A healthy diet requires plenty of fiber to feed our gut flora. In the same way here, I would say this is fiber for the forest gut flora. So we also see saprophytes interaction with symbionts, with symbiotic species, as the saprophyte break down the deadwood and bring nutrients from it, from the litter to the fermentation layer, where the symbiotic species can then absorb it into the tree network. Also, EM fungi, IFE, can also colonize coarse woody debris once in contact with the soil, and this can be an important source of nutrients, but also moisture for fungi in drought, during drought, as they act as sponges. And droughts are becoming less uncommon in Ireland in recent years. One final note on these fruiting bodies themselves, the fungi fruiting bodies are an important food source for wildlife like squirrels, deer, specialized insects, they all feed on them. In the picture here again, a oyster mushroom, Pleurotus austratus, Pleurotus pulmonaris here, both from Rostura here in Galway, munching away happily on, I think, willow deadwood. So just nearing to the end, just recapping a few overall considerations from what we discussed. So the EM fungi, ectomycorrhizal fungi, interdependent relationship with trees, it's clearly evolved over a long time, over millions of years, in fact, and it's a critical component of forest diversity and forest resilience. The symbiotic fungal networks are the foundation on which forest ecosystems are built, and they are essential to forest trees for sourcing key nutrients, as we discussed, but also pathogen control, water regulation, as well as supporting the very, very vast soil biota. And within the forest structure, species diversity, large mature trees, ectomycorrhizal trees in particular, irregular structure and deadwood are likely to strengthen the ectomycorrhizal network. This is a late addition, very, very interesting new project I came across only a couple of weeks ago. as part of my involvement with the Coforth Forest Ecosystem Services Group. This is Rhizocore, is a new UK-based biotech company that seems to have achieved what I certainly thought impossible, and most of my colleagues would have thought impossible, to culture symbiotic species and be able to suspend them in a tablet form for use as an inoculum at planted stage. They're now collaborating with Quilter Nature's team on a specific trial, and this project will aim to assess the performance of different EM local fungal species in boosting tree performance. And this is a project that I look forward to follow with great interest in years to come. So to conclude, as often with these things, we end up with more questions than answers. So definitely many questions still remain to be answered. And here is just a sample of a few questions that I put up as a catalyst. So what tree mixtures work best for productivity and resilience? How does the size, number and distribution of mature trees impact forest productivity, biodiversity and regeneration? How is the forest carbon budget affected by different forest composition and management? What is the role of EM fungi in driving these processes? How can close to nature management strengthen the EM fungi networks? And critically, we always conclude with more research needed. What are the research needs in this area in Ireland?
Great, thank you, Jonathan. I think there's no questions from people. We're very engaged with watching what you had to say. There's a question has come in now from Vanya. Do you know if Coforth or anyone else doing another or new ongoing diversity survey?
I am not aware myself of any.
Is anybody else maybe at this meeting who might know? I mean, if you do know, you can turn off your mic and jump in. No,
what I might be able to say that I was involved in a consortium bidding for European funding with some Scandinavian partners to bring such a project to Ireland. We weren't successful, but it might come around again. So there's probably some work underneath the ground on this, underneath the surface on this.
Okay. I have a question about the whole EM. So if you're looking at the evolution of maybe like a pasture to mature forest, does the AM come first with kind of the herbaceous and the smaller trees, and then you get the EM as the trees get bigger? Is that the tension between those two? Or do we know?
I don't. That's an interesting question. I don't have the answer to that. I don't know. I know that through the millions of years, species have come and gone, and the symbiotic ability of fungi to work with trees has been redeveloped in history over time. So it hasn't always been there. It's come and gone. But I wouldn't be able to answer that question fully. No.
Okay. So Edith's wondering the slides be available. So if you're happy for me to send out the slides to everybody who's registered, I can do so.
Sure. Yeah.
Yeah. So I'll send everybody who registered on Eventbrite. I can send them out, and I'll take the recording and I'll upload it to YouTube, and I'll send you all a link to that as well, so you can kind of see the live thing as well. So if you got here late, you can catch up. So there's some questions here now about what happens when you clear fell.
Yeah, that's an interesting question. There's studies done specifically on that, and the EM species would remain alive for some time. They will be completely gone after 3 years, but they persist for some time. So there are some ways to rescue, say, and to minimize the loss of the species by immediately planting. A clear fell site is highly recommended, and this is also a recommendation that I think comes to bypass some of the pests, the pine weevil problems with spruce. So planting immediately after clear fell would be advisable, but they take about 3 years to completely die. Yeah.
So another question, is there any point in spreading the spores of mature Botulus edulis in a 17-year-old oak forest to stimulate predominance of this as mycorrhizal species? And what are examples of EM shrubs?
I don't think so. the new fungi patches are very slow to develop from spore. They actually propagate mostly from the mycelium itself spreading within the forest. No, I don't. You can try. I know some people have tried on a large scale to do that using sort of a fermentation batches of fungi in water and trying to see whether that would lead to the propagation of species, but I don't think it's very effective.
And are there any EM shrubs? Are they mostly a big tree?
No, they probably are. I don't have the full list here. I can't think of any. Probably some of the willows can share on both networks. There's some species that they kind of straddle across. So I think some of the willows possibly, yeah.
Okay. So birch are pioneers and also EM trees. Could there be a fungal spore in the birch seeds, small as they are?
Could there be a fungal spore in the birch seed? I don't know. That's a fascinating concept, whether they could travel together. But again, the germination from spore, as far as I understand it, of new mycelia is very slow, and it won't be immediate. I have experience over years, because I've been visiting the same forest for nearly 30 years in Ireland now, and I know where they are. From a foraging perspective, I know them individually, which tree they grow with and which patch they are. And I've been surprised in finding new patches appearing here and there over the period. So while they can be born from spore, brand new baby fungi, if you will, they're actually, the main root is from mycelium extension, I think.
Okay. So somebody says, Tonya says, some shops sell mycorrhizal fungi with trees. Is it ever a good idea to add them at planting?
I think that's is commonly done for truffles, which they are a symbiotic fungus. And a lot of research went into cracking the code for truffles because they're highly valuable. So most of truffles to date are actually harvested from planted orchards across the world. So you can readily buy truffle inoculated trees for planting, as truffle orchards. I'm not aware of trees inoculated with other species of ectomycorrhizal fungi, but it definitely would be an interesting idea. You will equip your tree with a life support system right off the bat. So a nursery, I've experimented with this over the years. It's just proven too expensive over time to scale it up. So most nursery wouldn't use this, but certainly would be a great benefit.
Okay. Does the ash dieback fungus impact below ground EM networks?
Well, as I was saying, ash is an AM species, so it does not share on the ectomycorrhizal network. If it was planted in mixtures with other species, it wouldn't have an impact. I mean, the fungus just attack is a parasitic fungus. They attack the tree ability to grow and survive. So it wouldn't. I don't think it would have an impact on it directly.
Okay, so any tips?
Again, if there was anybody in the audience that have some insights in this, this kind of cutting edge research,
absolutely, yeah, jump in.
You might jump in, yeah.
So we have a question from Nick Hill. You mentioned the importance of pioneer species, but any other tips for EM promotion and woodland creation?
Well, I refer to that project, new biotech company. That's amazing concept that I never thought it would be possible. If that was to prove successful, that would be definitely Rhizocore. Basically, like you were suggesting, the birch seeds will come with their own packet of spores. You could, to prime an open field with ectomycorrhizal species, that would be an excellent way, and probably cost effective, because they would save, at least the idea behind it, they would save on fertilization, would save on replacement of trees, it would save on grass maintenance and so on. But I think the pioneer is what nature would do. The pioneer species is the one that come first in open terrain where the fungi aren't there, and they just have a way to cope best with it, while at the same time culturing the pioneer species, the fungal species. So I think pioneers in mixtures is the way to go.
Okay, thank you. So Sean O'Connell wonders if it's possible to map these underground networks and their evolution as they and the tree grows. Any simple methods or recommendations?
Any techniques that I've looked at through the research, they're extremely complex and dangerous as well, because you're looking at radioactive material and whatnot. So I don't think there's a simple way. If there was a simple way, we would have studied fungi and their networks long ago.
Yeah.
It's only with advanced technology coming into play and becoming affordable that we're now getting a better look into it. So I don't think it's for the amateur to look in detail into the network.
So,
but sorry to
yeah.
interrupt. One way we can have a proxy is looking at the fruiting bodies. So to observe in the autumn as they show themselves. By the way, the species throughout a year, just there might be fewer species of this fungi throughout a year. At any month of the year, it just they seem to surge, say from August until now, let's say.
So you would recommend as a forester that you would go and visit your woodlands and really note the fungi that are fruiting and get to know what they're called, and keep a record of that so you can tell what's happening.
I think that's a very simple way, and it's a fun way if you want.
Yeah.
It's to get to know your forest and your species. And once they realize that those aren't just sitting on the surface and there's nothing underneath them and that's it, there's like a curiosity. There's a lot going on behind the surface, and there just is the reproductive organ or the fruiting body of a much larger and more significant organism.
And is there a reference book that you would recommend for figuring out?
Excellent question, because there's a lot of books out there on fungi, and there's nothing worse than a book that doesn't have a broad range of species, or at least it's important to be aware that a book that has 20 fungi or 40 fungi is missing 1,000 odd fungi. So if it's not necessarily inside your book. So there are very good books. I don't know if I'm at liberty to put them out there, but using scientific books, it's very important, fully comprehensive.
We have one guest sharing the book with us here, I think.
All right, Aina's got Roger Phillips' Mushrooms. You recommend that?
Excellent book, yeah, I would say. Look, if we are putting out names, there's another one that is unfortunately is out of print, but is the Collinsfield Guide. All I would say is mushroom and toadstools of Britain and Europe. You're talking about thousands of species, maybe between 1,000 and 2,000 species to have a chance of having what's in your forest in the book. Okay, so you need books with a great diversity of species in it. That's probably the best way to describe it.
Okay, and then there's apps that I recognize. Go ahead.
Could I just add there that it kind of helps if you try to kind of find somebody, and mycologist somewhere in a group that you're with or that you know, because even when you think you found something, sometimes you didn't notice that the pattern on the stem is slightly different than what you thought it was, or something. You know what I mean? It could be something as simple as that.
Subtleties, yeah.
Oh, no, look, thanks for this. Thanks for the interjection. Absolutely, books are very dangerous things. In just alone, I absolutely would advise, if you're very interested and determined to learn about fungal diversity, find a mycologist, find someone that had at least experience, if not studied it, because it's a very complex matter. It's one of the hardest things I had to do, is to learn how to identify fungi, forest fungi.
Right.
Yeah, I think many of us spend many times and enjoy field days, field trips, field events, studying silviculture, timber production, development of our forests, for many things. I think there's a great audience out there for field events on mycology. And it's maybe something we should be all more engaged with. Jonathan, I think Sean's question in Leitrim might come from, I think you were tantalizing people there with your video of nutrient flow, of the underground nutrient flow. So I think everybody can imagine they can go out and start surveying the below forest floor network there. Fascinating to see.
So there's still lots of questions here. Shall I keep going through them?
Keep going. I mean,
yeah.
up to you.
Okay, so there's There's one about, given the fact we have a significant proportion of monoculture Sitka spruce that is in its first rotation in Ireland, would these forests have substantial EM networks yet? Does it take a long time, large trees set before they become interconnected?
It's very site specific, soil specific. I found a high diversity of species in even young Sitka spruce plantation, but the caveat there is they were planted on older sites with maybe fragment of oak forest. And what happened there, what I reckon it happened, that the diversity jumped across to what is actually a very strong ectomycorrhizal species being Sitka spruce. We don't have studies done specifically in Ireland on this. I know that from a kind of a greenfield side, there were studies done in British Columbia where they're trying to answer this question: how long does it take for the ectomycorrhizal network to come back from being wiped out? And they found it took decades, 20, 30 years. So maybe that's the time frame. Nature takes time to evolve. So maybe 20, 30 years is what it might take for just full development, but it's very site specific.
Right. So there's an interesting question here, which is close to my heart, about Suzanne Simard describing her motivation to save the ecosystem of the Canadian forest, which was been destroyed by glyphosate spraying of birch trees seen as weeds. So obviously the glyphosate killed the EMs, and the birch trees benefited the growth of Douglas firs. So are there ways in which your own work is able to reduce the use of harmful plant killers such as glyphosate in Irish woodlands?
Well, if a tree is growing healthily, it wouldn't need it. There's no need. In fact, the general advice is to reduce the use of herbicides anyway. In general, aside of any discussion on ectomycorrhizal fungi, they are, I wouldn't say phased out, but they are certainly not encouraged. One thing we always say is that if we give a good start to a tree, getting good stock, ground preparation is good, the tree is planted at the right time, you might need any weeding at all. So that's the optimal. And if you factoring maybe the ectomycorrhizal dimension added through addition to pellets, might become normalized in the future, or through the introduction of nurse, pioneer species that would help accelerate the culturing of the ectomycorrhizal network, herbicides might become not necessary, or even, so that's the way I would frame it.
Okay, very good, thank you. Any research done on the growth of second rotation forest where EM networks may be present versus the initial rotation, specifically in spruce?
Well, I would say hot planting, as it's called, just planting immediately. Another thing that can be looked at is retention. Individual tree retention is possible, so this needs to be planned ahead. And especially, it might be difficult as the forest is in full flight, let's say, is mid-rotation, is going, but say coming back again, planting blocks of different species. This one was minded trying to maximize or optimize the ability of fungi to help us grow trees. You could plan retention within that and plant blocks of different species that would outlast the harvesting of the trees. And I understand that some of the new planting mixtures coming in the new program might go in this direction, facilitating the planting of such mixtures in blocks of species that might be longer lived. So there's different techniques and design approaches that we could factor in with the fungal network in mind as well.
And I suppose if people are thinking about, taking up on the new grants to have forestry in place, to work with someone like ProSilva, who will do things in a way that will be best conducive to both the underground as well as the overground.
Ultimately, it results in productivity and increased value from the forest. So it's not an issue of biodiversity. It's just strengthening our the performance of our forests. Looking at a strong ecosystem, we deliver results, being in timber production, being habitat, water protection, that the forest is healthier overall. So it's a big ally, I think, in our management, and maybe one that we haven't looked at closely enough. And it sometimes often comes out like in nutritional deficiency, or trees aren't growing, we don't know why aren't growing, and perhaps the answer is under our feet.
Okay, can overforaging impact future fruiting?
I don't think so.
Okay.
In fact, this is a point that you know, in some case, it's been put to me that even harvesting fruiting bodies could harm the organism. It's not my experience over 30 years that I forage patches. They're grown bigger and bolder for it. It is like an apple on a fruit tree. That's the way I look at it. In some respect, they want to be picked. What I do, I always bring with me open containers if I do collect species, so that I'm actually broadcasting the spores. And this is a standard practice around Europe. What I would be more concerned is damaging the soil and the network within the soil through compaction, etc. But I have no problems harvesting. One thing I would say, leave the small undeveloped specimens to reach sporing, maturity for spore, so they can fulfill their ecological role. But I wouldn't have any problem with collecting mushrooms in the forest. No.
Okay, fantastic. Is it worth leaving a good amount of thinning as deadwood in a first intervention in a new oak plantation?
It's always a good idea to leave deadwood, and the jury's out. And I know some colleagues, ecologists and foresters out there, might have different views on how much is enough. Deadwood is always good. You have to be practical about it. So I think so. We don't want to have a forest that is denuded of deadwood. You know, a super tidy forest without any deadwood sometimes can be seen as a waste of timber. But I see it as, you know, it's just fiber for the gut flora of the forest, and it comes back into productivity eventually.
Great. So Vanya's given us a few links, so that's worth people checking out if they want to scroll back to that. Thank you, Vanya. We got the identification, got that question. Okay. I think the plug question, I think that's going to be researched by Teagasc in the near future.
Quilcha is.
Oh yeah, right. Okay, sorry. Yes.
Quilcha project, yeah.
Another the same
Quilcha
question
nature.
there. We got the pesticide one. Okay, very good. There's a picture mushroom app worth getting. So maybe just, you know, I mean, I wouldn't depend on it, but just to even get you in the right direction. I'm
old school. It probably is. I have an experience. I seen one for wildflowers for flora, which I was amazed what he was able to do. I did hear there is one for fungi. I haven't used it. I'm old school. I just rely on my.
You have the knowledge already.
On my training, but why not? I won't be able to comment, but try it out.
Yeah.
Try it out. Yeah.
Okay. Very good. Let me check that one out. Does alder and similar species that can coexist with both EM and AM have a distinct advantage?
I would think so. It has to. It has to be. So the species that can go both ways, clearly a lot more flexible. And if you think about it, they kind of inhabit habitat. They're kind of shifting habitats. Alder, I'm thinking in particular on alluvial and maybe floodplains, and they're more shifters. And maybe that's what they had to do over thousands of years. I'm just speculating, but it has to be an advantage.
So Owen Sullivan gives us a reference to Coford's Forest Fungi in Ireland. It's a good book.
Yeah.
So an online free PDFs, that's definitely worth having with you when you go out, or referring to when you come back. Are the EM fungi tree species specific? Are they non-selective? I think you kind of answered that in the talk.
They can be either or. Some are generalists, and some are very fussy in what trees they want to target. So maybe they're just like ourselves, you know, we're different likes and dislikes. Some are more tolerant than others.
Fair enough. Somebody mentions that we should leave some of the fungi for the wildlife.
Oh, of course, yeah, no, it's yeah, no, absolutely. As I mentioned myself, they are food for wildlife. I consider ourselves part of wildlife in some respect. We're part of the ecosystem, but as far as it's shared and it isn't a pillage, yeah, I think we can take our share and leave plenty for wildlife, because, as I said, you know, a lot of animals and invertebrates rely on that for their food. So you're on mute, Anna.
You're on mute, Anna.
Yeah.
Thank you. Okay, great. So I think that's the end of the questions that I was able to see. Let's see if any new ones have come in.
Yeah, Kieran there. Kieran's looking to come in, Anna.
So,
yeah.
Sorry, I just despite the encyclopedic knowledge you have in this area, Jonathan. It strikes me that we're only really scratching the surface in the knowledge we have collectively as to what's going on here.
Yeah, that's a fair statement. We are late starter collectively studying fungi for obvious reasons. They're very difficult to study. But we're getting going. I'd say we're getting going the last number of years. We're new technology. Maybe like my last question there is what our research needs in Ireland. Would be great to see a research project on this, specifically perhaps on Brazilians and mixtures. We are just scratching the surface, so I would agree with you. But you know, we do know something. It's not like we know nothing about it. There's some pointers that I hopefully try to put there. So maybe there's more knowledge than most people think we have, but there's a huge amount we don't understand.
Okay, so I might just go back to this slide deck then and just finish this up. So I think we've done a great job on the questions and discussions. So thanks very much for contributing on that. And thanks for all the positive comments for Jonathan and for myself. Much appreciated. Thanks to ProSilva for putting this on. Really informative and really enjoyable evening. So for me, the next steps, if you don't belong to ProSilva, you certainly could think about joining. It's not very expensive. You don't need to have a forest to be a member, but it's a great community to be part of. And the link is on there. You can follow ProSilva on Facebook and know what's going on. And you can watch the previous webinars on YouTube if you're interested.